Welcome to another episode of the Dendroid Podcast with Said and Nika. This is a conversation with Dr. Morgan Fullerton. He is an immunometabolism researcher and director of flow cytometry and Virometry core, and an assistant professor in the Department of Biochemistry, Microbiology, and Immunology, Faculty of Medicine, University of Ottawa.
During his scientific career, Dr. Fullerton has investigated how the immune system's function is shaped by metabolism. His lab explores immunometabolism in areas including mapping signaling pathways like AMPK and mTOR in immune regulation, studying choline metabolism in immune cells and its impact on inflammation, and exploring the role of immune metabolism in diseases such as obesity, fatty liver, atherosclerosis, and insulin resistance.
We hope you enjoyed this episode, and now here's Dr. Morgan Fullerton. Good morning, Dr. Fullerton. How are you doing today?
Well, how are you?
Good as well. Thank you for asking. So, I'm going to start by asking you this question. Could you tell us a bit about your academic journey and what drew you initially into the field that you're studying right now?
Okay. It depends on when you consider the start of my academic journey.
Okay.
Well, I guess that is up to you.
No. The year was 1988. No. So I did my undergraduate degree in Guelph. And so I had started in a molecular biology and genetics program, and I stayed in that program for my undergrad, which is only interesting, but a lot of my friends, you go to university, you do a first year program, it's quite general, and then you just picked something off of a list for your undergrad, and then you always end up changing your major because you become interested in something else. But I just sort of stuck with molecular biology, only because I found it interesting, but it was really all of biology that was still, other than ecology. Ecology never caught my interest. But as I continued through undergrad, the third year came and went, and now it was time for undergraduate research placement. And I ended up picking a lab after many different labs sort of interviewed me and then decided maybe I wasn't the best fit for whatever reason. I ended up joining a lab who did phospholipid research, or research into phospholipid metabolism. And I think I've said this story many times that had it not been for the fact that I chose that lab, I think my research could be very different. We could either be having a different conversation right now, or we might not even be speaking, just because of the decisions that were made past that choice. So I started in a lab really just interested in research. And I was very lucky that in my third year, very similar to TMM, I guess maybe the initial TMM laboratory that was in place, not the C phages, but I was able to take a course that was really focused on lab techniques. So for 2 full semesters, I was in a really comprehensive lab course, and it was just nice to be able to see variety. You weren't just pipetting something that was a restriction enzyme, you ran it on a gel and then check, you write out the lab report. It went much deeper than that. So I thought I came into fourth year pretty prepared for whatever research was going to throw at me, and I think I was. At the same time, I wasn't necessarily caught up in what I was doing in terms of what it all meant. And only that means that I enjoyed the process, but I didn't really know why I was doing it. And it wasn't until probably 15 years later that I looked back and was able to sort of piece together what it all meant, because then some of the research questions that I'm asking now really started way back then. But all that is to say that I picked a fourth year research lab that really did set the trajectory for what I ended up being interested in, if only for the point that it was like the guiding compass. And I could have been pointed in very different directions and maybe it would have all worked out. But from there, it was a matter of then finishing the undergrad, going to the grad program. That the project that I was working on really lent to characterizing a knockout mouse. It was a wide open project. It could be anything I really wanted it to be within the data, but it really pointed me in the direction of metabolism, and then that sort of interested me more and more as it was like a feed-forward process, which is a weird way to, it's only scientists use the term feed-forward process. But yeah, I enjoyed it, and so I have also said that I was never really cognizant of what I would do with the degree that I was working towards, which is never something that I never advocate to students right now to say, just stay in research if you like it. because it'll all work out. There needs to be a little bit of forward thinking, which I'd never had, but I knew I enjoyed what I was doing, which was, for me at the time, the most important thing. And so then that led to a postdoc, which we can get into if you want, and then from there, even at that point, even after I finished my PhD and was comfortable taking the next step of training, even though people say, oh, if you do a postdoc, which is what you would do after a PhD in a research setting, that pigeonholes you, or that focuses you and narrows you down to only be able to enter into academia. I think that is probably the biggest misnomer in all of academic sciences for undergrads through to PhDs, is that a postdoc can only lead to academia, and if you want to do anything else, postdoc is not for you. Because for me, I just thought of it as a guaranteed job, which is something that's actually quite nice to have coming out of a 5-year degree after having a 4-year degree after 5 years of high school. It's nice to think that you're going to have something that pays a little bit more than just whatever being a student is. And so for me, it was a nice way to enter into that. And without being a student, it was actually quite freeing to say, all right, if you didn't have classes, if you didn't have TAing, if you didn't have tuition, what could you do from a research perspective? And I was very fortunate to be able to align with a researcher who has just opened his lab at McMaster, and the projects were the perfect time that I could come into them. And with the experience that I had and with the questions that we were asking, I developed new, I guess, expertise or experiences that then really I was able to do the work there that then ended up in the papers, or as a paper format, was relatively impactful, we hope. They were important enough questions, they were published and well received, that was when I think I decided, at this point, I don't want to do anything else. I'd like to continue doing this, and if I want to continue doing this, trying to be a professor is the way to go. And so it wasn't as though I was always focused on that, which I know people who were from the beginning. They took undergraduate high school or undergraduate biology. They knew they wanted to do this. I wasn't there, but when I did come to that point, I was fortunate to be able to interview here. I started in 2014, and yada, yada, yada. Here we are.
Makes sense. I think also one of the points that you touched on, that like the things you were studying in your undergrad later came up and you understood what it meant. I think it's also very similar for us too, because I remember in biochemistry we had a lot of things that we had to memorize and learn, and as we are going, I go back and like, oh, that was what that meant. Like, it makes sense.
Exactly.
Yeah.
Like even in the lab, there's a lot of things that we must do from the techniques, and well, if you just want to keep the pace in the time, you want to just get it done in the time frame that you are given, you can't just do it. There's no time to look back and, okay, so what exactly did I do? What just happened?
Yeah, and I was very focused on, all right, I'll do the protocol, which meant, yes, of course, reading the protocol the night before, writing it out, but why and what the big picture of that wasn't clear. And yeah, it wasn't until I was sitting down to write my insert grant that I thought, oh, I did this before, and if that happened, but then I did something else, if that happened, if I put those two things together, that's a question that forms, and that was one grant. So
makes sense.
So what was the research question that directed because you mentioned that the project that you were given or you found,
mm-hmm.
That you aligned at the same time for your postdoc were like the perfect, so like all the stars aligned.
Yeah, because the supervisor that I worked with was in Australia at the time, and so this person had done their PhD in the same department that I did my PhD, but he had left before I got there. And so there was a little bit of lore about this person, and that they had done quite well. And so when I emailed and then set up a phone call, knowing that then I had the job, I think what perfectly aligned me to that lab was that the project that I worked on in my PhD really just opened up into a project on whole body metabolism. Like it focused on one knockout, the knockout of one gene in a mouse. And so if we want to understand what a gene does, we take it away in a mouse and then ask the question, is this mouse normal? And it turned out that this mouse particularly, when it didn't have this gene, or when it didn't have enough of this gene, they became obese over time. And so that led to a whole bunch of metabolic complications, which then I had to learn, what does that mean? How do you test for that? What are you measuring? But with all of those base skills, that's what I could apply to the questions that this other researcher, so Greg Steinberg, I worked with in McMaster, he was asking sort of analogous questions of what happens if we take away this protein and this sort of process, and are the mice normal from a metabolic standpoint. And it just so happened that the experiment that they had set up, that I really had no say in at the time, but again aligned my direction pretty well, was that they had taken a mouse that lacked one gene in the whole body, but they'd taken the bone marrow from that mouse and put it into a normal mouse. So now it's only the bone marrow that has the knockout. And so they were specifically asking, from an immune context, does having or not having this particular gene in the immune system going to affect the readouts after we feed them a high-fat diet? So now these mice become more obese the more high-fat diet they eat. They become less and less responsive to hormones like insulin, and they become more and more hyperglycemic, which is more towards the diabetic side, and there's more baseline inflammation, which is important because inflammation should be there in the context of something acute and it needs to fight off an infection. But when situations that are more chronic and metabolic instigate, then the low level of inflammation there all the time is actually a bad thing. And so it was that initial study with the immune system, with inflammation, but having metabolism be the driver of that, which really, I think, set me up to then. A lot of people at the time were just coining the phrase immunometabolism, and so I kind of just lumped myself in with that, and that's about how that started.
Makes sense.
And is that what you're continuing with the same direction?
Yeah, we sort of finished that, but then from the answers that you get from one study or one question, you get 100 other questions that then you dig down on or dig into. So for me, the answers that we got from understanding what happened to this whole body physiology when the mice lacked this certain protein in the immune system, the whole body physiology was really from the lens of type 2 diabetes and obesity, whereas cardiovascular disease is very interrelated, a lot of common risk factors, but the presentation of cardiovascular disease usually happens within the arteries, but underlies a lot of the same dysregulation of metabolism. So there's dysregulation of metabolic pathways, there's dysregulation of inflammatory pathways, and so the same model that we had asked the question of, is this important for obesity and obesity-induced inflammation, I then wanted to ask the question, is this pathway, are these models, can they be used to ask the question, is this important for the progression of atherosclerosis, which then sort of precedes things like heart attack and stroke. And so I worked on that in my postdoc a little bit, but that's primarily how I started my lab. Those were the questions that I had asked preliminarily in my postdoc, and then got a few exciting results, or enough to know that I was on the right path, or at least this was a question worth asking, and that ended up being my first CIHR grant when I started my lab, is asking the question, if we took away a specific metabolic regulator in a specific cell type that's important for atherosclerosis, is that going to tip the balance one way or the other? Are we going to make things worse? Are we going to make things better? And then we kind of played around with that, because then if you're going to take away something, we could always add it back or use some sort of drug to try to stimulate that pathway and ask the same overall questions of whether or not it makes a difference for disease.
So how do you identify the gene, the regulatory gene, to begin with?
That's a good question. I think that nowadays, and I am always envious to a certain degree, but I'm in awe of people who ask big picture questions of the difference between a situation where we have a healthy situation or metabolic dysfunction. And there's a big push now to then screen from a multi-omics perspective. We want to not just ask a tunnel vision question, which is what we're very used to, is asking the question of what is the consequence of taking this one gene out. Mm-hmm. There's always, probably for the last decade and a bit, there's been a push to look in an unbiased manner at the beginning of a study, or at least that's how they're presented in the papers that we read in journal clubs, right? Where you see the RNA-seq analysis that was done between a control and a disease state, and then based on the most highly upregulated or most highly downregulated genes or proteins, those are what people focus in on. I think that is a great way to, in an unbiased manner, to really focus in on what could be different in a discovery manner. We haven't done that. We did the opposite, which we because it's now 2025, there are thousands of papers being published every day. A lot of people, not everyone should just start off by asking this big picture screening question. So because we know so much about all of these pathways already, I've been of the mindset that we can still ask the specific questions on what, even if we're the only ones who think that they're important, I think being able to understand how they're important in the context of the bigger picture and being able to convince others, because if it's just us to believe it, maybe it's not worth believing. But the proteins that we're looking at, or the regulatory networks that we're looking at, there are central players in that, and those are what we're trying to focus in on to ask the question. Because if a central player is important, then understanding how a master regulator of metabolism, let's say, and what that means is vague, but it just does something. Protein in a cell does a lot of things to regulate one aspect of metabolism. I still think, and probably will take a pretty strong stance on this hill, that is worth understanding. And there's just so much to understand, because as soon as the question becomes different, is this important in neurodegeneration? Is this important in cancer? Is this important in atherosclerosis? There may be underlying similarities between those questions, but they're all having different outcomes, and so they're all worthwhile questions, and they could all be from a very specific standpoint of, is this one protein important for that, yes or no? And then that's going to tease out more understanding of the pathways. So it's just kind of a bottom-up or a top-down approach.
So like the other type that you mentioned is like, well, there is this problem. We do a bunch of analysis, and these are the potential culprits or causes. Then you take that and you knock it out, or you do the stuff that you do to actually make sure to confirm the causality that yes, in fact, if we do that, this is what it causes. And potentially, the next question would be, okay, then how?
Yeah. So that could be, and so we've kind of been guilty of not taking that journey. We've kind of hopped into that pipeline at the point where we have the target that we want to ask the question about, not necessarily doing the initial question of what might be different and what might be overarchingly causing one phenotype. Because it'd be interesting to talk to, even in this setting, the author of a paper who took that approach, because I think we've all read the bigger papers out there now with Science and Cell, where they present that type of trajectory, and they've gone through and they found 3 different genes. They knocked out those 3 different genes, and then it turns out that they were important in the hypothalamus. So they knocked it out in the hypothalamus, all as part of one paper. That's a lot of work. It takes a lot of money. And so I think the having that plan, having that pipeline is an amazing accomplishment, but it's not attainable for everybody. And so I think that if we can kind of carve out different studies that I can cumulatively put together an idea of how these pathways are acting, and I think more importantly how these pathways are underpinning how some of the therapies that we use now that we know work, but we just don't know how they work. That's where I think having targets already in mind of asking, is this target, does this protein mediate the effect of this drug? Because if we knew that, all of a sudden now maybe we can make this drug better by understanding how it's interacting with this one protein or this one signaling axis. So
I mean, the previous literature is there for a reason. We can just, that's a good starting point.
Yeah.
What are the common pathways that come up as you study like different diseases and immunometabolism?
I think there was the common pathways that we look at.
Yes.
And so it really is from the lens of, I mean, metabolism is such a broad term. So I could be convinced that, or maybe I could convince anybody that every cell is metabolic. So when we say metabolism, it's what we're talking, I guess the importance of metabolism for us is through the lens of a metabolic kinase that is sensitive to the amount of energy that is in the cell, the amount of nutrients that are in the cell, the amount of actual stored energy in the form of ATP or these adenine nucleotides, AMP and ADP. The protein that we're looking at, AMP activated protein kinase, it physically binds some of these nucleotides that when energy levels are low, so when ATP levels are low, reciprocally we get a high level of AMP that then binds to AMPK and actually turns it on. And when it turns on AMPK is a kinase that will phosphorylate different targets. And it's sort of like, I mean, I know you guys know this, but for the audience, if you needed to turn on a light, turning on the light switch is akin to phosphorylation. It happens really quickly and the outcome is very quick. If you needed to turn on the light but the light was burnt out, then all of a sudden maybe you have to go to the drawer, you have to then change out the light bulb. That's maybe akin to regulating protein, different levels of proteins, right? If we needed to all of a sudden then say, well, we don't have a protein to regulate metabolism, we need to make it. And we already have the transcript, so we just need to translate that and get the protein. That takes longer, but it's still faster than having to find out that you're out of light bulbs, go to the store, come back, then change it out. So that would be like, we don't have the RNA, we need to now transcribe the RNA to then translate the protein. So all to say is that phosphorylation is quite quick, and because these kinases are what are doing the phosphorylating, this protein AMPK can turn on and turn off a lot of pathways that all told, all together, will either boost energy in the cell or turn off things that are taking up a lot of energy. So protein synthesis, lipid synthesis, those are things that are really energy demanding in the cell. And so the first validated pathway that AMPK was known to regulate was turning off lipid synthesis. And it does that by phosphorylating two proteins in the cholesterol and the fatty acid synthesis pathways. And it has a dramatic inhibitory effect on those pathways. And so the effect is very quick, which is what it's supposed to. And then on the flip side of all of that, these are not the only two metabolic regulators, but for us, the two that are important because of how they reciprocally regulate metabolism. So AMPK can be thought of turning on catabolic things, catabolic pathways. And so that means that if they're going to either suppress energy use or generate energy through different mechanisms. Whereas the mechanistic target of rapamycin, which is mTOR, and there's two complexes, but we're interested in complex 1, so mTOR complex 1, is a protein kinase that really phosphorylates and regulates pathways that are the opposite. So this will stimulate protein synthesis, this will stimulate lipid synthesis. It's all about now with its sufficient nutrients, with insulin being a hormone that's pro-anabolic, all these anabolic pathways are being upregulated in response to turning on mTOR. So now we've got a kinase that's turned on during energy deprivation, so AMPK, so maybe during fasting AMPK is high, and then during feeding AMPK is low, but mTOR is then high. So there's this reciprocal regulation. When one is up, the other is down, and we know that they can communicate to each other by phosphorylating and inhibiting each other. So my rambling was only to be able to say that we've now asked specific questions with this signaling axis and others for if you have a metabolic kinase that phosphorylates 100 different things. And so if you take out that metabolic kinase and then you ask the question, is this important for obesity? And it is, that's a great answer, or you now have the answer to that question, but you are no closer to understanding why, because you have now affected 100 different things, because the thing that you've taken away does 100 different things. So with the power of mouse genetics and using CRISPR, we've been able to make point mutations on the target proteins of these metabolic kinases that specifically cut the wire. There's inputs from the metabolic regulator to each of these downstream targets, and we've been able to change the serine that becomes phosphorylated to an alanine to then take away the ability of that protein to be affected by the kinase. So now all of the other 99 things that kinase does is still functional, and we can ask the specific question.
Sealing that one switch.
Exactly. Just cut the red wire. Don't cut any other wire but the red wire. And to be honest, a lot of the times, the answers are pretty confirmatory. These are things that we have known that targets can be phosphorylated by kinases for decades. And there have been phosphoantibodies that have been generated to be able to understand, is this signaling axis different under different conditions? And the answer can be yes, the answer can be no. But then people likely jump to the conclusion, if there's an increase in the phosphorylation of a target, they say that is mediating the effect. That must be why. If I see evidence that this kinase phosphorylates this target, and then this happens down here, it must be because this target is phosphorylated. And I just think that that's a flaw in the logic. And so if that would be, if that was written differently, if the word associated was put in there, I would be completely fine with it. But they haven't actually shown mechanistically that phosphorylation is important for anything. And so, with these CRISPR models, with these serine to alanine mutations, we can now ask those questions that may or may not confirm what people have always been saying, maybe conflating in the literature, but for rightly or wrongly, or for better or worse, we are asking those questions and trying to find answers as to whether or not these signaling axes are important. And in the end, we have evidence that this type of approach can be impactful in terms of it. This can yield better understanding of how a therapy that has been used for generations actually mediates a beneficial effect. So that was work that I had done in my postdoc. It really set up the stage that we can use these types of models to understand those types of questions.
Makes sense.
So from what I understand, it's to just fill in that gap.
Yeah.
That it's just an assumption that so this must go through that.
Exactly.
Actually, does it or does it not?
Yeah.
So I guess if it does, as you mentioned, it's confirmatory, it confirms. What happens when it doesn't? Then.
And that's been the case sometimes, and so now I still think that those are worthwhile. I think I've said this before on other forms and fashions, is that we have probably more often than not in my lab proven the null hypothesis, which means hopefully no one goes into an experiment with an expectation, but you can have a hypothesis. And the hypothesis usually is that there will be a difference. And that's fine, because you're going to prove or you're going to disprove that. And if you prove it, great, everyone loves a difference. And then you can drill down on how that difference is mediated or why that is fixed. But if there is no difference, that is a valid answer to that question that I think a lot of people just dismiss, because in the end it's not as colorful, it's not as well received as differences that are in big papers and publications. So we've made it a habit to publish the stories that the question is there a difference when we do this one thing? And when there isn't a difference, that's worth reporting. And so this, while it may be confirmatory, or it may be, I don't know, the opposite of
I
confirmed.
think it's even more interesting because, if you already, if that is the assumption and then the assumption is confirmed, nice.
Exactly.
If it's not, that means, that you should look there to see, if it's not doing that, then what else is happening?
Exactly. And I think that's, for us, in the particular example that I had for my postdoc, we used a knock-in mouse that AMPK could not phosphorylate the very first substrate that was found as protein that mediates fatty acid synthesis. And so the mice, when they're fed a low-fat diet, there was a phenotype. There was a difference in the mice. And we think that's because as we made this metabolic change, the change was different enough, there was enough of a difference that it mediated a phenotype, an effect. It wasn't visible, but when we looked at the livers and when we measured things in the markers in the blood, we could see a difference. Where we lost that effect is when we fed the mice a high-fat diet. And so now, if we superimpose obesity onto this phenotype, the phenotype goes away. The mice were completely indistinguishable in all of the same markers as they were between genotypes. And so for us, we just thought that yes, obesity masks the effect of this pathway when all of these nutrients and fats are present. But it then provided us an opportunity to intervene with a therapy. And now we can specifically test if this therapy is known to, or is thought to, work via a certain pathway, including the pathway that we've now disrupted, we have the perfect model to test if it's important for that therapeutic benefit. And it just so happened that it was very important for this particular drug's beneficial effect. And I think that we've contributed then to the understanding of how metformin works from a fatty liver and from a diabetic perspective. So I think those are the paradigms and the sort of study designs that I think that we will continue to superimpose, and the question will just maybe shift. I mean, we're asking questions in atherosclerosis, we're asking questions in infection and type 2 diabetes and obesity, and they all have this sort of common links of the metabolism that links them and the inflammation that in the end will always be there. So.
It's just fascinating how these pathways mediate information in one way or another. So phosphorus, as you mentioned, but it's a switch, and it would either it's a signal, but it's also chemical, how information is being like propagated chemically. Yeah.
Another molecule that comes up frequently when I was reading was choline. Where that fit into the play?
So that was what came out of my undergrad.
Okay.
And so I only say undergrad because it was right before I started my master's. It was the summer before I actually started my master's, in between my research project and my master's. And I was finishing up a project that a student in the previous year had started. And choline is a nutrient that we can't synthesize enough of it in our bodies, so we need from our diet. And the lens that was the PhD lab that I was working in was specifically because they were interested in one choline transporter. So this nutrient choline needs to be taken up into the different cells. It can't just diffuse through. And so the result that I had in my pre-masters was really that as we differentiate monocytes to macrophages, so an immune cell that's circulating in the blood, to its terminal cell type is a macrophage, which is tissue resident. This choline transporter, the expression goes away. From the surface, it comes off of the surface of the cell, and the expression goes way down. And then, I guess, in terms of the importance of that, it was really just that I then knew through my postdoc training that if you activate a monocyte to a macrophage, the cells go from being quite small to being bigger. So that automatically means that you need more cell membrane. One of the cellular fates of choline is that it gets made into the main phospholipid in the cell, phosphatidylcholine. So PC is what we call it. So this phospholipid, you can't make a bigger cell if you don't have more phospholipids. And by virtue of it being the main eukaryotic phospholipid, it didn't make any sense to me that this choline transporter, which we always expected or hypothesized that the transport of choline into the cell was for phospholipid synthesis, it can't be going away and going down if we were needing to make more lipid. So those are the initial questions that I think I revisited and sort of questioned why and what we actually showed in these first publications, to then ask the question, or I think I came to the realization that a lot has been known about choline as a nutrient. You guys heard of Charles Best and Frederick Banting? So Banting discovered insulin along with Charles Best. So anyway, they're best known for, no pun intended, for the discovery of insulin. But Charles Best continued his work and showed that if you were to take away choline in dogs, they get fatty liver almost within 2 days. So there are links with choline and metabolism outside of just the cellular metabolism in terms of phospholipids being important for everything. So fast forward to have my lab, I have these initial questions that really have to do with sort of an immune context. Is choline metabolism important for a model immune cell type for us, which was a macrophage? And with that question in mind, it was always, we were able to ask it from a little bit of a more unbiased way, so a more general, is choline metabolism important for inflammation, or is inflammation important for mediating or for changing choline metabolism? So those are two reciprocal questions, which, if I'm going to be honest, were only really being asked because the question that we were trying to ask, which was, is this one choline transporter important for these things? We were having a really hard time validating the knockout model that we were trying to use. And so we didn't have a genetic model to work off of, and we then ended up sort of asking these reciprocal questions, which were nice because they were reproducible. We could superimpose on a macrophage that we took from a mouse. So we would end up harvesting bone marrow. We'd convince all of the bone marrow to differentiate to one cell type. It's an artificial, but at the same time, the end result is a very nice way to get a lot of macrophages that are very similar. And then we could split those macrophages into 4 groups, and then we could what we call polarize them. So macrophages are very dynamic and plastic. They really respond to what's in their environment, and they change based on what they see. If they see something really pro-inflammatory, some sort of bacteria or viral component, they will get angry and they will try to fight. All of their responses are based on what they see from their environment, or not necessarily they get less angry, but there's different types of inflammation that end up being less pro-inflammatory and potentially more regenerative or pro-resolving, because the inflammation sort of goes up and then we need to resolve the inflammation. So the macrophages can be important for both. But with all of those sort of tools in a cell culture comparison, it was nice to be able to then sort of superimpose the question of choline metabolism, because the tool that we would augment, or the change in the condition, is the inflammation, but the readout is choline, and how choline gets taken up into the cell, how fast, how much of it, what are the different transporters that are on the cell surface, what are the transcript levels, the protein levels. So everything about the readouts become about the pathway, and inflammation was the test, the instigator of potential change. And then once we sort of figured that out, which was that as much as most things change, I guess reciprocally with inflammation. If I was to say to you pro-inflammatory, what's the opposite of pro-inflammatory?
Anti-inflammatory.
Anti-inflammatory, right? And so those are how people usually describe the two sort of polarization states that we use. There was a pro-inflammatory and there was an anti-inflammatory. And it could be said that the anti-inflammatory should really be called pro-resolving, but they're two opposite ends of an inflammatory scale. But when we superimpose both of those effects onto macrophages, choline metabolism, when we had a pro-inflammatory situation, went up. And so I was anticipating that as if we have an anti-inflammatory situation, it would go down, but it went up to a similar extent. And what's interesting is that it's to a similar extent, the amount of phospholipid goes up, but the reason why is different. There's a transcriptional response that is, we think, specific to this one transporter that I was again working on in my PhD, and that we've tried to knock out. I'll get to that in a minute. But there are transcription factors that bind in response to inflammatory signaling to the promoter of this gene that in the end upregulate the transcript, upregulate the protein, upregulate choline import, upregulate phospholipid synthesis. So it's all coordinated, likely because similar to as a cell, a monocyte is differentiating to macrophage, these cells become activated, they become bigger, they become they phagocytose particles, so they need more memory. It all made sense. On the anti-inflammatory or pro-resolving side of things, it was really interesting because the protein level, the transcript level doesn't change, but the protein level does, and the choline uptake does. And so we think there's a translational effect there where the transcript is there, it's not changing, but there is an upregulation of the translation of that transcript in response to that situation. So nature's found a way to, if we can't find the right transcription factors to bind with the promoter to get the actual upregulation of this, has evolved a different mechanism to then arrive to the same outcome, which I thought was pretty cool, and I don't think I've come across very many situations or examples of something that will affect or have the same effect and the same effect size and the same direction with two what are considered opposite stimuli. And then from a reciprocal question, when we asked in the end, if we limit choline, if we take away its availability, it's a nutrient, so if the cells don't have it supplemented to them, then they don't get it, which is akin to a genetic knockout, right? We ended up not being able to use a genetic knockout. It's really hard to knock things out with siRNA in primary cells, especially macrophages, which is why we didn't use sort of an acute genetic knockdown approach. But we had pharmacological inhibitors, we had antibody inhibitors, and we had the ability to take away choline from the media. And when we did that, the cells got more pro-inflammatory when we stimulated them with a pro-inflammatory response. And there were very specific things that were different on the anti-inflammatory or pro-resolving side of things. And it sort of directed us to a very specific signaling axis where if you have IL-4 is the cytokine that you give to macrophages if you really want to test them in a culture situation to be this pro-resolving phenotype. The highest or the most upregulated IL-4 stimulated gene is a gene called RALM alpha, or retinal A or RALM alpha. It's a cytokine, and it was completely suppressed when we took away choline or when we used inhibitors. And so it's this interesting and still unknown mechanism as to why choline metabolism why altering choline metabolism affected this one readout. And so, but then we were forced with, we had a readout that was different. We had this huge effect on a difference in the cytokine. And there wasn't really a difference in any of the other typical cytokines. Sometimes if you think about inflammation or other things, you profile things, there's a a signature. If you're looking at differentiation of muscle cells, you could look at the most highly upregulated genes and call that a signature. With the signature that should have been there for IL-4 stimulated macrophages, everything else was the same, except for this one thing. And I often regret that we didn't go back and figure out why, but instead we sort of asked the question of, okay, if this is different, then what does that mean? Can we find a situation? Can we superimpose a condition, some sort of in vivo relevant paradigm that then we can then tease out a significance for this pathway? And so we collaborated with somebody in UC Riverside. So Mira and there, again, a quick email, and we had set up a collaboration. It was now then a co-authored publication. It's just one of those feel-good stories where it was just super easy, and they were awesome. Sam Young did the work there with Payman, who's a postdoc in my lab, so it was a first author for them and co-submission for Mira and I. But in the end, an in vivo situation where IL-4, which is the cytokine that we used in the dish, is relevant are parasite infections. So different model organisms, they're called helminths, so it's an intestinal parasite that we can then infect in the mice, and the result is a dramatic upregulation of IL-4 and all of the sort of in vivo consequences we could test by using an inhibitor of choline uptake. When push came to shove, there was no difference in the amount of control of the parasite. So if you ask the question, is this pathway important for controlling parasitic infection? The answer is no. But it was encouraging for us because we could, in the context of this infection, all of the IL-4 stimulated pathways go up. So this realm alpha is the cytokine that we saw is completely suppressed. That goes up in vivo, but when we inject it with the inhibitor, it goes down in every tissue and every immune population that we looked at. So there was a lot of links from the cell culture work that was then in vivo, even though there wasn't the effect that we had maybe been hypothesizing that we would see as a mediator or as a consequence of this difference. It was encouraging that we could see that there was still a difference. So reproducibility is huge within a lab, between labs, between years. So to be able to then see in an in vivo context that if we use the same inhibitor in intestinal crypts, those macrophages do not express any realm alpha, whereas the controls do. The question of whether or not that's important for parasite infection, well, the answer is it's not, but it's not to say that's still not important for something else. And so the only other thing I'll say is the other observation that we had was that when we used the specific inhibitor that blocked choline metabolism, there was a complete shift in all of the immune populations that were in the peritoneal cavity. So this was a peritoneal injection of a drug, so we think that there's sort of a local effect that might have been different if we were to give it systemically via the intravenous system, but in the end, this dramatic change in the immune landscape then has me thinking about, well, then is there a situation where this particular signature, this change, the downregulation or the upregulation of all these different immune populations, can we find a situation where that particular, signature could be applied in a beneficial way? And the only way I can really think is it's taking macrophages and it's taking immune cells that are more prone to being regulatory and pro-resolving, and it's skewing them to being more pro-inflammatory, which could be beneficial in terms of tumor microenvironment, where a lot of the times the immune system has been co-opted and has been muted and blunted, and people are looking for ways to turn back on the immune system. And so I'm always wondering whether or not this might be one of those ways. But future work.
So touching on the thing that you said, how are the other ways that we can translate your research into clinical settings to resolving diseases? That was one of them you mentioned. Also, I think you mentioned metformin as well, that you have provided more cues, but what other things?
I think for me, using the potentially translationally relevant drugs or therapies that have been, discovered at the bench, tested in preclinical models, so mouse models of disease and other preclinical avenues, testing those along the lines of these models that we've generated, so these targeted knock-in mice. If we wanted to ask the questions of, is this pathway important for the beneficial effect of fill in the blank. I think that's the easiest way to kind of translate that type of research. But that's also pretty painstaking, and a lot of questions that it's maybe not the best use of time, because while it's interesting and while it might be a nice side project, or it might be the culmination of a bigger question, I don't think that it's necessarily the way forward, because we already know these things work. So from a drug discovery perspective, what we're doing isn't necessarily as impactful as, let's say, starting from scratch, looking at the difference between two different conditions, finding a target that's upregulated, and then finding an inhibitor for that. And then I think those are still the avenues that are best from a target discovery and drug discovery perspective. But from a rare disease perspective, and from even just understanding, I guess I'm flipping up. Within my lab, we have sort of the metabolic regulation side of things, and then the choline nutrient side of things. We know of, in the world, there's probably, we now are collaborating with clinical collaborators who have identified patients who have homozygous null mutations in the choline transporter that we have a mouse model that is specifically knocking out this one choline transporter. And there's always been questions in my mind as to the importance of, if we've identified now, we being science, has identified 5 different choline transporters. One of them is specifically important for acetylcholine synthesis in neurons, and it's very validated that is specifically important for that. And if you don't have it, the animal doesn't survive. Whereas to get choline into all of the other tissues in the cell, it's still kind of an unknown question as to which transporter is more or less important. And I don't really care which one is more or less important. A lot of people, I think, conflate the importance of the gene they're working on with the relevance of it biologically. I'm just interested in knowing one way or the other. And recently there have been newer discoveries on choline transporters that hadn't been known previously. And so we'd always sort of thought that there was one family of which the transporter that we were working on / knocking out is thought to be the main one expressed, the main one for mediating this effect. Now there are others, and so it really does set up the situation of, it may seem competitive, but it's more potential for different things to be valid, or different transporters to be impactful for different situations. But I mentioned it translationally is because there have been patients identified with these mutations, and they present with a really early neurodegenerative phenotype. And so it's a rare disease, and so how many in the world we don't know, but we have patients, we've fibroblasts from 4 patients that span 3 different mutations that can go anywhere from a truncation mutation that's loss of function through to complete loss of any protein because it's a null transcript. And so with these, we have fibroblasts that we've been able to re-engineer or reprogram to inducible pluripotent stem cells. And with these stem cells, we can now differentiate them into different lineages. These are human cells. They can be differentiated to, still in culture, but into neuronal populations, or microglial or astrocyte populations, the populations in the brain that we would never get to understand in any type of cell culture situation had we not have access to these. And so it really brings to the forefront the ability to say, right, in these cells, can we CRISPR it back? Can we repair these cells? And then was this defect in this choline transporter, what was it responsible for? We can do gene therapy, so we can provide this transcript back with an adeno-associated virus or with a lipid nanoparticle that has the mRNA, just to really, again, if you can test the importance of something in science by taking it away, the further test to that, like the nail in the coffin, the perfect and ending to that experiment is, if you've taken it away and there's an effect, give it back and it should correct. It's a rescue experiment. And so those are simple rescue experiments, but I think that they actually have quite impactful potential for affecting maybe a very small percentage of the population, but that's, I think, the closest that we've gotten to a translational impact in my lab, which other people at the faculty in the world, a lot of people do that type of work, but our projects have always sort of been more on the preclinical, let's work to build the foundation of understanding. But now with this project in particular, it's yeah, it's quite exciting. And now we have a PhD student who's ready to drive that work. We have a knockout model that is hopefully going to be validated. It'll be interesting to see how it goes. And if you guys don't ask me about the problems that went along with that mouse, I won't tell you. But armed with a knockout mouse, which has the potential to look at the knockout of this gene or this transcript in a tissue-specific manner, as well as a whole body to mirror the patients and the iPSCs from the patients, I think that we are perfectly aligned to then work towards improving the quality of life of these patients, which I think, again, not everyone's going to get the chance to do, but I'm excited at where it might take us.
That's super interesting. I'm looking for that to come up.
Me too. It's still at the start, but
I'm sure.
exciting.
Makes sense. All right, now switching gears a bit, let's focus on your teaching aspect of life. Do you prefer teaching or research?
Do I love teaching or research? Or
Which one do you prefer?
Is there an equivalent to the Fifth Amendment in Canada? I prefer research.
Okay.
From the strict perspective of if I was given the chance to only teach or only do research, I would pick research. So the either or situation, I pick research. But from a teaching perspective, I do enjoy the teaching, where I think it would be interesting to get feedback from people candidly would be the course that I teach is molecular biology. You have taken it.
Yep.
So my approach to teaching is, I think, a little bit different than some other people's approach, and I have an understanding that it is different, and so there's a learning curve in the teaching and in the acceptance of said approach, but at the same time, I have a very finite bandwidth / window of understanding, and so my way is kind of how it is, and then people, I think, are better for being exposed to that way of thinking. I think it's bringing a lot of the research and the troubleshooting aspects of research and trying to apply it to a third year type of situation. The problem is, and I've said it before in many classes, is that if you're teaching a class that when you're teaching a particular topic is the first time students have ever seen it, I think that there's an impactful way to teach it for the first time, but it lessens the ability of how in-depth you can go when there are fundamental concepts that need to be understood first before it can kind of be applied to more maybe complex thinking. So I've always found that a little bit limiting, because as much as I would love to be able to dig in deeper to some of the concepts and to really have it be more engaging and problem-based learning, the fact of the matter is we need to go over what splicing is and how transcripts are spliced and what alternative splicing is. So I hope that there's a good balance, and I question every year sort of whether or not everything needs to be blown up and redone to be able to then really reimagine how the course should be delivered. But every year I come back to the fact that no, I think small changes and small tweaks are better than a huge massive blow up. So I think the question was which I prefer. So I prefer research. I do love teaching because for as much as my style is a little bit more open-ended, and I definitely don't advocate for, or I don't necessarily require people to memorize. It's actually the opposite. I am of the mindset that if you need to know something, you look it up. And the enzymes in glycolysis, I do not know them. I know some of them, but not the order of them, because if I need to know, I'll look it up. And so that's the approach, is really how can you integrate what you know and what problem solving and how can you think outside of a normal situation where you may be programmed to think that there's one answer, but in the end there are infinite ways to get to that answer. And if you can think creatively around the very linear way that you like, the path of least resistance is the one that I hope no student takes, because then it was always interesting to read answers to a question, which they're all long answers, which everyone will complain about at the end. At the beginning, people complain about, but I guess the rewarding part is to read an answer from a student that is something that I would have never thought of. But as I read it, I'm thinking, yes, this could work, or it's improbable, but it's still possible. And I don't know, it's very rewarding when I'm reading these answers, and then more so at the final lecture or there's been lots of grumbling during the whole year, and it's thank you, I really learned a lot, or this was different, and it was, not everyone does that. Some people just give you cut eye and leave, but in the end, I like that feeling. So
I wasn't into class, but like all the stuff that you just mentioned, I like
You've
got
heard,
it secondhanded, like from people coming out of the class and like the projects and the exams.
Also, being in your class, I actually really enjoyed like the method that you teach, because it forces you to be creative and like use the things that we've learned during the course and apply it into a problem that we face. It is challenging, yes, but it's very also rewarding. Like you feel very good after the exam. Like I think that kind of makes sense. Like if you go through that route, and also it applies for like further in life. Like if you're like writing the grants in the future that we are also doing for some of the classes that we have right now, that helps a lot.
I mean, I hate to break it to everybody listening, but you're going to fail 9 times out of 10, and it is okay to be wrong. I think that was the one thing that even at the graduate level, it's hard to convince students to be able to offer up their opinion or their insight into something, because there's this fear of being wrong. So it makes it difficult when it's an undergrad situation. There's 60 people in a class and you're asking people, they have no idea, they just want to be told the answer, but you will understand the answer infinitely better if you get it wrong and then understand how you went wrong. You're programmed to understand it better if you make a mistake. And I think it would be different. I hope it's better that there isn't the approach, because the third year, as you guys entered into this program, it's different than second year. Again, I give the intro lecture, third year is always different than second, but this program or the way of teaching in the particular program that you were in, it was different than what we hoped it would be classical teaching.
Mm-hmm.
And I then superimpose an even bigger change onto that by asking you to think outside of the memorization box and propaganda and think up a creative answer. And in the end, because I know how hard that is and how big of a change it is, and I'm not saying just do this and stop whining, I'm hoping that there's a little bit of empathy there that goes a long way to then getting people through it, because in the end, certain people like it better than others, but I think it will serve everybody better in life.
Makes sense.
So what do you do outside the lab?
I go home.
That is a great start.
So I grew up here in Ottawa, but really only from grade 7 on. So it was growing up in Ottawa wasn't necessarily now living in Ottawa, but I enjoy sports, but don't follow sports anymore. I have 2 kids that in the end take up most of the time, but we enjoy being outside. Rock climbing is one of them. The kids are still trying to find their passions. So I grew up playing soccer and basketball and volleyball, and I would love to play soccer again, but it's one of those things where I'm now teaching and coaching soccer rather than playing soccer. So if anybody listening has a team, I can play. So, one thing with academia, I will say, there's no, it is a pyramid scheme, to be honest. And sorry, I'll get to the point in a minute here of why I'm saying this, but you start as a high school student, then you go to university, then you go to grad school, then you go to postdoc, then that is the pyramid aspect of the professional researcher type of situation. So I was very fortunate to be able to make it to here with a combination of privilege and hard work and timing, but along the way, there are certain perks that inevitably you get to lean into. So what I'm talking about now is I don't have a hobby of traveling, but being able to travel as part of one's job outside of being constantly gone or travel being stressful is really a huge benefit to this job. And it's, again, something that we're extremely privileged to be able to do. But I remember going to, my first conference was in Banff, and then I went to San Francisco, and my partner came with me to San Francisco. She had a conference in Paris. I came here to Paris, then we went to London for a day, we came back. And then at my postdoc, we went to Greece. I went to Palm Springs, and then took some time off, went to Thailand. And then when I started my lab, I went back to San Francisco. So the traveling aspect of it, for better or worse, it's really nice. And so, again, very fortunate to be able to make that happen, even to be able to bring someone along. But now that the kids are older, I had a conference in Australia 2 years ago, and I was always silently / very vocally pressured into being able to take a sabbatical and go to Australia to live there. And I dropped the ball on that. At least so far, I've dropped the ball on that. But with a conference in Australia, and I was an invited speaker, we decided to go for a month. And so that travel as part of the overarching lifestyle it's very nice. And you can, travel is one of those things where I wouldn't say I traveled a lot, but I kind of just listed off everywhere that I've been, plus or minus a few. But it just gives such a bigger appreciation that if there is, in any capacity, the ability for anybody to travel and to experience different cultures or spots of the world, again, through an academic lens, is a very privileged one, but it's something that I'm glad that my kids are being able to at least be at a point now where they can remember it as well, and hopefully it can have an impact.
Nice.
Yeah. If you weren't a scientist, what career would you choose?
I forgot about that question. I'd often say during my PhD that I would be the most educated construction worker, because I'm handy enough, and the problem with that is that I don't have the engineering prowess to then be able to do things probably properly. Mm-hmm. With YouTube, I'm sure everyone can do everything, but I'm relatively handy, so I think that I would enjoy being in construction. And maybe we've just undergone a pretty big home renovation, and I had some pretty critical input at various stages that I was very proud of myself for being there, for having a very good suggestion that in the end made it better. So I'd probably
being the fix-it man.
Yeah, I could maybe do that. I enjoy, as much as this sounds a little bit weird, I might be able to be a sous chef. I don't cook often. I can cook, but only in so much as I can follow a protocol. But my wife and I took a cooking class, and it was interesting, more so to see all the first dates going around, and we had been married for a few years, so it was funny, but I learned how to chop properly, like to hold a knife properly, and because I had always, then I really just, it could be that we cook, and so I would prepare everything for her, and then she would be the one to put it all together, and now I'm the one also preparing it and putting things together, so I don't know, I like cutting stuff, so maybe I could be a sous chef, but also being a professional retiree or a rock climber would be awesome. Like seeing different lifestyles, I think it would be interesting to be able to settle down and to do something completely different, that if you could, and if it was part of a business. I often think that there are, have you ever been to altitude climbing gym in Gatineau?
No, but I've had students who go there.
Okay. They converted a church, an old church, into a climbing gym.
Oh.
And so I can now just superimpose that business model onto every church that I see. And it might ruffle some feathers, but that's what I think I would use those types of buildings for, is just to be able to then introduce that sort of...
I mean, you are transcending in one way or another. So.
I think so. You're coming back down, hopefully, but yeah, I think that would be kind of cool too.
Makes sense.
What are your favorite meals to make after, like the before and after?
Also, he cooks a lot, so he always asks that question.
I mean, I really wanted to. So we went to Thailand in 2011, and I love Thai food, but I can't do spice as much. My wife loves spice, can do as hot as it comes. It just doesn't work for me. It just doesn't. And if it eclipses the taste, I just can't. Right? There's no point. So green curry is my favorite, but it would always be too hot. Always. And so they didn't have enough milk for me to drink along with the curry. And so I thought to myself, it would be really nice if I could make a green curry for myself to be able to regulate the spice. And so I would say Thai food is what I've gotten into. And so I'm making more curry pastes and growing the Thai basil myself. And so I enjoy that probably the most. But I have been able to branch out, and at least there have been recipes where I'm always like, oh, I want to try this. Like, I'll make that for you. And then it ends up being a special thing, because I'm not as hopeless as to not be able to follow the recipe. And I've made some silly mistakes, but always test the potatoes first. don't just go with the fact that they seem good. Test the potatoes, because nobody likes a crunchy potato.
Makes sense. Also, you've been in different institutes, as you said, like McMaster's, Guelph, and also you Ottawa. Which one out of the three, or even if there are more, which one out of all of them do you prefer?
They're all different. I think you only appreciate them after you leave. Mm-hmm. And so Guelph, I was a student. I couldn't wait to get out of Guelph. But looking back on it, Guelph is a city block in a small town that goes to, I mean, at the time it went to 100,000 during the year, and then down to 60 in the summer. Like, it was a university town, but it was all in one campus. And so there was definitely a sense of you were on campus during that time. And then for grad school, it was still at Guelph, but again, it was living close enough to the lab that I could bite there in 30 seconds. And so it was just a lifestyle that was different than then being able to own a house in Hamilton as part of my postdoc. My wife commuted to Toronto, and so it was a different feel, because now I was in the lab. I dropped her at the GO station at 6:10 in the morning. I was in the lab by 6:30. I picked her up at the GO station at 6:30 at night. Like, it was work for a good few years, and it wasn't until we ended up getting a dog that changed. But Mac was nice because it was that phase. So if I had transferred similar to sort of a 2-year undergrad in one and 2-year undergrad in another, I think I could probably juxtapose them and compare them.
Mm-hmm.
But Mac was perfect for where I was at that time. Guelph was perfect for where I was at that time. And I interviewed one other place that in the end I could have envisioned myself being academically. Spoiler, the Oilers are a good team, so I could have cheered for the Oilers, but it's one of those things where Ottawa trumped Edmonton I don't even know that I would have gotten that job. I interviewed there, but I don't know that I was getting it or not. So when it was a question mark of where to go, I think I was very fortunate to be able to have Ottawa on the list of places that wanted me, and Ottawa's a perfect big small town for me. I don't need or want Toronto or Montreal. Those are too big for me. But Ottawa, it's central enough that I can run to work, bike to work, walk to the grocery store, and yeah. But I've probably been, I've used the line, I've been to the main campus a handful of times. In the end, it's now a bit more than a handful, but this is the University of Ottawa for me. This building, which I just told you, it's the only second time I've been in the library. I didn't even know it was here until last year. So it's all about perspective. And so from my perspective, I go to my office, I sit on my computer, I maybe go into the lab, because it's right off of my office, but it's really hard to compare across the board. And I think that it's better that way, because as you diversify your experience, then each of the places that you go can add to your overall life goals, trajectory, research experience, life experience. That if you could probably get all that experience in one place, but as soon as you switch, that difference in perspective, I think, is going to be pretty powerful for giving you the tools that you might need to move forward. So, yeah, Ottawa right now.
Yep. Well, that's a pretty good answer again for right now.
For right now, yeah.
We should be asked the difficult question.
You might as well. I will probably skirt around it. But.
Okay, so what do you think is the meaning of life?
The meaning of life. So, I won't go into the research part of it. I think if you can, that question comes with a whole bunch of privilege and situational awareness, that I don't want to say happiness.
Mm-hmm.
But at the same time, I think that's probably what people could strive for. It might mean different things to different people, whether or not it's family, whether or not it's self-fulfillment in terms of relaxation or professional meaning and self-worth. I don't know. For me, I guess it's family first, and then I still love what I do. I've told people this story that I wrote 2 grants from scratch last March, and I was up till 2 a.m. for a long time, just trying to get it done. And at no point did I think to myself, I have to be up till 2 a.m. It was just still an enjoyable process. And I think that told something to me, that I'm very lucky that funding has come through. But in the end, that process is quite stressful and drives a lot of people away. And it has yet to affect me that way. And I think I still love seeing people come into my lab and be able to succeed, and to have a situation that maybe wasn't optimal, and then all of a sudden it clicks and their abilities just sort of multiply and exponentially increase, and they start in the lab with nothing in terms of experience or expertise, and they leave an expert, and more so than I. And so all of that to say is that the professional satisfaction that I have right now really just sort of helps to complement the work-life balance, that it needs to fluctuate. And so the happiness all comes from the work-life balance and the life being pretty good and work being pretty good. And I'm very fortunate, and fortunate for the good people on my team that are making it that way, and for the family who put up with me.
Nice.
So,
I think that sums up everything. Thank you very much for your time today.
thank you for having me.
Thank you.
Thank you.
Thank you for listening to this conversation with Dr. Morgan Fullerton. If you're interested about his work, you can find the link to his website in the description.
This is a Dendrite podcast. To support it, please share this episode with your friends and anyone else who might be interested. We hope you enjoyed this episode, and see you next time.